Hi everyone, welcome to the tutorial on membranes and cell organelles. Today we're going to be looking at what a cell membrane is comprised of and the various functions of the cell organelles present in plants and animals. So, the cell wall, this is the boundary between the cell's internal contents and the external environment. It allows the cell to maintain a stable internal environment. It is comprised of a plasma membrane. Okay, this plasma membrane is fluid. Okay, and we'll get to this in a second. Okay, but mainly it is a barrier preventing the crossing of substances. Okay, selective permeable barrier. It maintains an internal environment for each cell. The role of the plasma membrane: it keeps substances out of the cell. Okay, it prevents the passage of substances that it doesn't want to cross. Okay, selective permeability. Okay. However, it can allow the passage of other substances either through channels or through diffusion. It also has many receptors on the surface of the cell, allowing for it to receive signals to assist the cell to do certain functions. It also assists in cell-to-cell communication. Alright. Structure of the membrane. It's made up of a phospholipid. A phospholipid has a phosphate head and a lipophilic tail. Now what this means is that the head is attracted to water, okay, as it is polar, and the tail is lipophilic, okay, so it is not attracted to water, which means it will pace inwards. So it's a double membrane. Alright, the phospholipids point outwards, and the tails point inwards. The lipophilic tails point inwards. Okay, this creates a barrier of water. Alright, so the water can't pass through. However, it does dissolve into the surface of the membrane. Alright. Now, the membrane can move laterally, up and down, giving this fluidity. However, if the membrane was just completely composed of phospholipids, it would be very easily broken. Right? So the way this phospholipid it keeps its integrity and fluidity is the adding of other substances such as proteins and cholesterol. Right? So in the membrane, we don't just have phospholipids, but we have steroid lipids, such as cholesterol in animals and phytosterol in plants. Okay, this gives it the membrane its structural rigidity and fluidity. Okay, and it's an ability to maneuver or move in response to the environment. There is also glycolipids, right, the combination of a carbohydrate and a lipid molecule present in the surface of the cell, and they serve as cell surface markers. Okay. So other components include glycoproteins, right, carbohydrate and a protein, and these usually act as cell surface receptors. Alright, so they're important for cell communication, cell-to-cell communication. Alright, and various other proteins. Now these proteins in the cell's membrane are usually signaling proteins, or they're usually to do with being cell channels for components of to cells through the cell membrane. Okay, so like ions, or glucose, water. Alright, various channels to allow substances to pass through. Okay, the membrane proteins consist of transport proteins. These are allowing substances to pass through the membrane. Receptor proteins, okay, these allow hormones to bind, alright, and signal the molecule or signal the cell to do various functions. And recognition proteins, they act as antigens or they act as cell surface recognition markers, which tell the immune system that this is your self cell or this is a foreign cell. Okay, there's also adhesion proteins, which allow cells to link together, which is very important for eukaryotic organisms. So here we have an example of the cell membrane, alright, with a cell surface receptor protein, a recognition protein, a transport protein, and adhesion proteins, okay, in the roles within or embedded within the phospholipid membrane. Okay, and as you can see, cholesterol, very important molecule for structural rigidity and fluidity of the membrane, is dispersed throughout the phospholipid bilayer. Okay, this allows the membrane to become fluid and structurally sound. So the fluid mosaic model, okay, of the cell membrane is all about the fact that the cell membrane can move like a wave, kind of, okay, and this enables it to adapt to changes in its environment, so being pushed or being maneuvered. Okay, the mosaic part is in reference to the protein channels and the other molecules that sit embedded in the membrane itself. Okay, so cell organelles. Alright, within plant animal cells there are various organelles that proceed to do various functions, and we're going to look at them today. So first we're going to start off with the control center of the cell, the nucleus. Alright, Within the nucleus, there's supercoiled DNA, and this DNA is there and it serves as the instruction for the cell. Okay, now this instruction for the cell will be different for every cell depending, it's the same in every cell, but different sections are methylated depending on what the function of that cell is to do. Okay, so because not all the DNA is transcribed and translated in each cell. Okay, so the nucleus comprises the house of the DNA. Okay, it also comprises of the nucleolus, and this is where ribosomal RNA is produced. All right, this is where the RNA is produced to create ribosomes. Okay, so the nucleus houses the DNA. It's the site where transcription occurs, not translation. Translation occurs at ribosomes, which we'll get to in a sec. All right. Okay, so the mitochondria. Mitochondria are known as the powerhouse of the cell. Okay, this is where all the cell's energy is produced, the ATP for the cell. Okay, it is a double membrane structure, okay, creating two internal environments. One between the inner and outer membrane, known as the cristae, and the inner section of the mitochondria is known as the mitochondrial matrix. Okay, along the inner membrane is where the cytochromes sit, allowing respiration to occur, or aerobic respiration, or, as you should know, electron transport. Okay, this is where majority of a cell's aerobic ATP is produced. So, mitochondria, recapping, two membranes, internal, right, external. The space between the internal and external is known as the cristae. The space on the inside of the mitochondria is known as the matrix. All right, this is where majority of a eukaryotic cell's ATP is produced during aerobic respiration, during the stages known as the Krebs cycle, which functions in the matrix, and the electron transport stage, stage 3, which occurs in the membrane, or the inner membrane along the cristae. Alright, ribosomes. So, ribosomes can be found in two locations. They can be either found free-floating in the cytosol, or they can be found in or attached to rough endoplasmic reticulum. Okay, now DNA is translated, transcribed into mRNA. The mRNA leaves the nucleus. It proceeds to go to one of these places, all right, either the free-floating cytosol ribosomes or the ribosomes on the rough endoplasmic reticulum. Now, the ribosomes on the rough endoplasmic reticulum synthesize protein for export, and the ribosomes in the free-floating cytosol synthesize proteins for the cell to use itself. Okay? The rough endoplasmic reticulum, or the endoplasmic reticulum made up of smooth and rough. Right? The smooth is where lipid production occurs, and the rough is where protein synthesis occurs for export. Okay? It's known as the channels of the cell. It allows the cell to move proteins and lipids around to the various organelles, around to the Golgi complex to be exported. Okay, so the Golgi complex. This is the site or the organelle that exocytoses the contents of the cell into the outside world. It is linked to the ER, and the ER, or the rough ER, is a site for protein synthesis for export. So the ER synthesizes proteins and sends them to the Golgi. Often these proteins are modified further by the Golgi before they are exported. When ready for export, they are put into something called a vesicle, a double membrane structure that will subsequently fuse with the plasma membrane, allowing the Golgi to exocytose the contents into the outside environment. Lysosomes. They're unique to eukaryotic cells, the site of breakdown of debris within the cell and of external debris or foreign microorganisms that might be harmful to the cell. Okay, a cell that uses lots of lysosomes is a macrophage. Macrophages have lysosomes on the inside because they engulf foreign debris, right? They're part of the immune system. They engulf debris that comes in from the outside world or dead cells, and they destroy it via the use of lysosomes, enzymes built to break down molecules and debris. Vacuoles. Now, vacuoles are more commonly found in plant cells because they have to store their water for the long term. So a vacuole is a membrane space found inside the cell, alright, and it allows it to fill with liquid and store the liquid for the cell's use. Alright, you can also store food in vacuoles. You can also have contractile vacuoles and protozoa that expel water, allowing them to move. Okay, but usually plant cells are the ones where you find very large vacuoles, okay, and this allows them to fill up with water and keep the turgidity of the plant, or the structural support, solid so the plant can stay upright. Now, while we're in plants, chloroplasts, okay, are found in plant cells and some photosynthetic bacteria. These organelles capture the light energy and convert it into the chemical energy known as glucose. Okay? They're made up of a double membrane structure exactly like mitochondria, which is why scientists hypothesize that mitochondria and chloroplasts were once bacteria, known as the theory of endosymbiosis. This theory states that these were once individual organisms. This is backed up by the fact that mitochondria and chloroplasts contain their own DNA and ribosomes. Okay, so the theory is that they were once organisms that were then engulfed by other organisms and are now components of plants and animals, or eukaryotic organisms. Alright, and what they do is they allow us to, or plants, to convert sunlight into energy to store as food, which the plant can then subsequently use. The animals can then eat and burn through aerobic respiration in the mitochondria, allowing us to produce far more ATP to get to a higher order organism. So, the cytoskeleton. Each cell has its own internal framework, and this framework is made up of proteins that include microtubules, microfilaments, and intermediate filaments. The purpose of the cytoskeleton is to maintain the shape of the cell, right, and provide structural support. It also allows the cell to move materials around its internal environment. Now, these components of the cytoskeleton, microtubules are hollow, long filaments, right, made up of the protein called tubulin. Microfilaments are solid, thinner, and more flexible, right, and they're made up of actin. And intermediate filaments are very tough and made up of a variety of proteins. But their purpose is often to tie cells together, so to adhere cells to one another. Alrighty, so this is the first in a series of tutorials I'll be delivering. Hope you enjoyed it. Thank you.